Hello friends, welcome to my channel Clinical Biochemistry with Dr. P.K. Prabhakar. Today we are going to talk about DNA fingerprinting. This DNA fingerprinting we are also called as DNA profiling. Basically, this is a laboratory process which is known as DNA fingerprinting, and normally we are going to use the nucleotide sequence. Nucleotide sequence normally you might be knowing that we are having DNA, which is our genetic material, and this DNA is composed of the repetition of the nucleotides. So that is a polymeric unit of nucleotides where we are having adenine, thymine, cytosine, guanine is repeated. Now, this nucleotide sequence of a particular region of human DNA is going to be identified. How we are going to identify that I will tell you. So basically, DNA fingerprinting you might have heard about at many places. So some first before going in the details, just we'll see what are the different applications we can do it. I will tell you only four important application now, and later on we will talk about the detailed application of DNA fingerprinting. So if you'll see the application DNA fingerprinting application, this is going to be used in forensic sciences, where normally we are going to use to identify criminals or to detect some suspects from the crime sites and other places. And second important use of DNA fingerprinting is to identify the correct parent of a child or the for paternity testing. Then this is also going to be used for the diagnosis of many hereditary disorders, as well as like HIV or some other disease. We will talk in the last. And then another application of this one in the agriculture. So this is the common application because everyone might have heard about this DNA fingerprinting some or more time. What is exactly this DNA fingerprinting? If you'll see, rough on an average, about 99.9% of our DNA between two human beings are same. So we are having only differences in 0.1% DNA between two human beings are different. What is this 0.1% human being DNA, which is different among me or you or any two persons, if it is they are not identical twins? So this 0.1% DNA, normally these are the we are going to use to call it junk DNA, but exactly this is non-coding DNA. So by this DNA fingerprinting, we are going to use to measure to evaluate this 0.1% of the DNA. So it is a method used to identify the individual from a single sample of DNA by looking at the unique pattern in their DNA. And what is this unique pattern? This unique pattern is the repetition of some nucleotides. So in some cases, 3 to 5 nucleotide, 10 to 15 nucleotide, or some amount of number of nucleotides are going to be repeated many times. So that is called as unique pattern. This unique pattern of repetition is present in this 0.1% of the DNA. So it is a method to find the difference between these satellite DNA. This repeated DNA, normally we are going to call satellite DNA. Later on, I will tell you what are these satellite DNA, what are the different types of satellite DNA we are having. So these repeated units of DNA are called as satellite DNA regions, and that repeated sequence we are going to identify this DNA fingerprinting. Same amount of DNA can be repeated in two different persons, but the number, like in my case, the same sequence is repeated 10 times, it might be repeated 11 times in you, or 12 times in your brother, or 13 times in your sister. So the number of times, how many times it is going to be repeated, that might be different, or the sequence might be different. Now, the satellite DNA regions are the stretch of repetitive DNA which do not code for any protein. So they are non-coding sequence of the DNA. Non-coding sequence initially we used to call it junk DNA. So these non-coding sequences are the form a major chunk in the DNA profile of human being. And this repeated sequence of satellite DNA normally decides how or the difference between the two individual is because of this satellite DNA. So they depict a high level of polymorphism. Polymorphism means many times it is going to be repeated in n number of times and are the basis of DNA fingerprinting. These genes show a high level of polymorphism in all kinds of tissue, as a result of which they prove to be very useful in forensic study. This DNA fingerprinting was invented in 1984, almost near when we have a very marvelous discovery in the genetic engineering, that is PCR. So the DNA fingerprinting was invented in 1984 by Professor Sir Elec Jaffrey. After he realized you could detect variation in the human DNA in the form of these mini satellites. So Sir William Elec Jaffrey had discovered it. Now, DNA fingerprinting is a technique that simultaneously detects a lot of mini satellites. What is mini satellites? We are having three different kind of satellite DNA. I have told you. After this one, we are talking about. We are having mini satellite, micro satellite, and macro satellite DNA. So, this DNA fingerprinting is a technique which simultaneously detects a lot of mini satellite in the genome to produce a pattern which is unique for an individual, and this is a DNA fingerprinting. The probability of having two people with the same DNA fingerprint that are not identical twins is very small. So, like our fingerprints are not matching between the two individuals, similarly, DNA fingerprint of two individuals are similar. It is very impossible scenario is there, except in case of identical twins, there might be similar. Now, if you see in DNA, we are having a repetitive sequence, and we are having three types of satellite DNA. It is microsatellite, mini satellite, and macrosatellite. These three satellite DNA we are going to classify on the basis of their nucleotide regions, which type of nucleotide rich region they are having, or how many times if it is going to be repeated. So, microsatellite normally it is having repetition of 1 to 9 base pair, where in case of mini satellite repetition of 10 to 100 base pair repetitions will be there, and in case of macro satellite more than 100 base pair repetition will be there. Now this micro satellite is have most commonly it is AT rich region, so there it will have adenine and thymine rich region will be there, and they have roughly composed a stretch of 10,000 to 1 lakh base pair. At the same time, if you see the mini satellite, they, which is having the 10 to 100 base pair repetitions, they are normally GC rich region, and they are having 500 to 30,000 base pair repetitions will be there. And then microsatellite. And these all are called as short tandem repeats, STRs. Normally, this mini satellite we are going to use, or this is going to be applicable for the DNA fingerprinting, as we have seen here also. So, this mini satellite is the main things which we are going to evaluate, which we are going to use during the DNA fingerprinting. Now, what is the how this DNA first DNA fingerprint are going to produce, or how we are going to use it? So the principle is it is the combination of DNA sequence that frequently use differences between the people are identified by sequences. So short nucleotide repeats that are inherited and vary in a number from person to person are the most crucial need to be DNA fingerprinting. In this case, the first step is the DNA need to be extracted from the cells by doing centrifugations. So DNA isolation is the first step. So DNA is taken from the cell and purified via chemical processing and centrifugation methods. After that one, if DNA amount is very low, then we are going to increase the number. We are going to amplify it through polymerase chain reaction or PCR. So amplification if required, or after that one, these DNAs are going to be fragmented digestion by a molecular seizure, which is the restriction endonuclease enzyme. So the restriction endonuclease enzyme breaks down the DNA into small pieces. These enzymes cuts the DNA at particular locations, chopping it up into different lengths. So these are called as molecular seizure, which going to digest the DNA into different fragments. After this DNA fragment are going to be separated by agarose gel electrophoresis. Agarose gel electrophoresis normally separates DNA on the basis of E by M ratio. So depending on their size, the DNA fragments are next separated using the process of this gel electrophoresis. In the presence of an electric field, normally it is a gel electrophoresis or electrophoresis is a technique where we are going to use electric field and DNA are going to be separated. Not it can we are doing for protein also, we are doing for anything also, but so DNA fragments are positioned on a transparent gel bed on that is on the glass plates or for electrophoresis, which involves applying the electric current into the gel. After that one, these agents that separates the DNA fragment into single strands are then introduced. Then the separated DNA fragments are visualized by the UV radiations by applying the dye that is called as ethidium bromide. Normally, this ethidium bromide dye intercalated between the nucleotide strands, and so by using this ethidium bromide, we are going to visualize the DNA sequences. Now, the DNA is transferred to the gel electrophoresis plate to the nitrocellulose plate, nitrocellulose membrane or nylon membrane sheet. This is called as southern blotting. This repeated sequences, which I have called you microsatellite, these are called as VNTR probes, are going to be used. Variable nucleotide tandem repeats probes are going to be added, which binds at specific nucleotides that are complementary to these nucleotides, and this is called as hybridization. Once it is going to be hybridized, unbound DNA is going to be washed out, and the hybridized DNA fragments are detected by autoradiography by using X-ray films. So this is the process how this DNA fingerprinting going to be used. So if you'll say like we have collected a blood sample from a crime site, from there from these cells DNA isolated. If required, it will need to be amplified through PCR. Then digestion will be done through restriction endonuclease. Restricted DNA is going to be separated through gel electrophoresis. After that one, this gel separated gel need to be transferred to this by into the nylon membranes or nitrocellulose membrane through southern blotting techniques. Then it is going to be hybridized by VNTR probes, and then unbound things will be washed out, and then it will be detected by X-ray films. So this is the overall process how this DNA fingerprinting is going to be performed, or it is going to be done. Now, this DNA profiling. So modern day DNA profiling is also called as short tandem repeats analysis, and it relies normally on the microsatellites rather than the mini satellite which is used in the DNA fingerprinting. So microsatellites or short tandem repeats are the shorter relatives of the mini satellite, usually 2 to 5 base pair long. So in DNA profiling, normally we are going to use the microsatellite, not the mini satellite. In DNA fingerprinting, we are going to use mini satellites, whereas in profiling, we are going to use microsatellites. So microsatellites are rather smaller than the mini satellites. Like mini satellites, they are repeated many times throughout the human genome, such as example is given there. How this DNA profile is going to be produced? This is the steps. So DNA sample we are going to having. Then PCR prime labeling will be there. So labeling, then we are digested. After digestion, this is a short term repeats, then electrophoresis, and then we are going to visualize it. So this is profiling. Now how we are going to solve the crimes or criminal sites, or the from the. So in this case, DNA profilings are very useful in the forensic, because only 80 very small samples of human material is going to be left at the crime sites, and that may be sufficient to identify someone. Normally in the UK, different countries having different types of STR sequence they are going to use. In the UK, a complete DNA profile normally consists of 11 short tandem repeat sequences plus a sex determiner to confirm its profile from a man or human sequences. Now, all new profile includes an additional 5 STR sequences, especially in the Europe. So, along with this 11 STR, 5 additional STR is going to be used in Europe. In USA, they are going to be used normally 13 STR, short tandem repeat sequences. Many states are increasing the number of STR so that better results will come. Now, if you'll see here, a match between the crime scene profile and the individual profile identified to detect a possible suspect. Like this from crime site, the sample which I've received. So this is the DNA profile, STR profile of the sample from the crime site, and these two are the suspect. Now, if you'll see the match of electrophoretic pattern or the X-ray pattern, so this is matching with this one. This is matching with this. This is matching with this one. This is matching with this one, and this is matching with this one. So if you see the more matching and better correct matching with the suspect 2. So in this case, suspect 2 is the sample is matching with this one, so he is the culprit. So a match between the different crimes in profile indicates a repeat offender at work. The police may use this DNA evidence to support other evidence to help prosecute someone for a crime. Now, this DNA fingerprinting or DNA profiling also going to be used for linking blood relatives. Like we get half of our DNA from our mother and half of DNA from our father. Like if you see, this is the tandem repeats of the mother, and this is the STR for the father. Now, in the child, they have got one from the mother and one from the father. So child will have a STR from they are getting from both mother STR and the father also. So STR are therefore passed down from the parents to their child offspring. Like you can see in this example, from both parents they are having the child is having STR from the both the parents. So DNA profiling can be used to help confirm whether two people are related to one another or they are not related. DNA profiling can also be used to identify victims of crime or a major disaster. We are going to detect some whenever some accident happens. If someone is not going to claim body normally to identify a persons or a victims that who is relative of that person. So there also this is going to be used. So DNA profiling has a high success rate and very low false positive rate. Now, if you'll see the application of DNA profiling in different fields in the details. So, first application in the forensic sciences. So, biological samples such as blood, hair, saliva, sperm, or different other body fluid cells are used for DNA profiling, and it is possible to compare to the DNA recovered from the evidence sample by using this variable number of tandem repeats, that is called VNTR prototypes. It adds into the investigation of crime like rapes or the murders or some other things. Then second important application is the personal identification. Like in this case, it makes use for an idea that DNA fingerprinting can be used to identify people, acting as a kind of genetic barcode. Then determining paternity and maternity. A person accept their VNTR from their parents. Like we have just now seen example that child gets from father also and mother also. So cases that where the disagreement have been resolved via parent-child VNTR prototype analysis. Additionally, immigration and inheritance proceedings may also use these informations. Then another application we are having in the breeding programs. Breeders normally typically assess a plant or animal's genotype using this its phenotype. Since homozygous or heterozygous dominance is difficult to distinguish between appearance, the genotype can be determined using great care and accuracy using DNA fingerprinting. Then we are having diagnosis of hereditary disorders. It can be used to identify inherited disorders in both newborn also and the prenatal child also. Like some of the disease where we are going to use this one is cystic fibrosis, hemophilia, Huntington disease, then Alzheimer's, sickle cell anemia, beta thalassemia. So many diseases we are going to use this DNA fingerprinting profile to identify in case of prenatal as well as the newborn babies. Then the creation of the treatment of inherited disorders. DNA prototypes linked to the disease can be identified by examining the DNA fingerprinting of family members who have the history of particular disorders. And the last one is the detection of HIV. So in that case, like you can see, the person with HIV can be diagnosed by comparing the HIV RNA bands, which converted to the DNA via RT-PCR, reverse transcriptase PCR. So we will get the DNA from the for the HIV with the bands formed by the human blood sample. So this is a number of applications we are having, but commonly most commonly we are going to use for maternity three, that is maternity, paternity, forensic science, and diagnosis of diseases. There are, when we are doing performing this DNA fingerprinting, number of issues can come. Like sometimes you are going to get DNA mixture, sometimes the degraded DNA will be there, sometimes restriction enzymes does not works, or some STR short term repeat does not works. So number of things are coming. So whenever issue comes, that time we are normally going to solve it. Otherwise, this is a very good applications. Similarly, we are going to use genetic markers also, like we are having RAPD, RFLP, AFLP. These are the different genetic molecular markers we are going to use for the identification of different application of polymorphic DNA. So this is all about for today. Hope you have understand it. If you have any query, any comments, you can write in the comment box. If you are not subscribed my channel, you can subscribe it. Thank you very much. Have a nice day.